With Milo from ProteinSimple, you can perform single-cell westerns and measure protein expression in thousands of individual cells in a single run, good for validating single-cell RNA data at the protein level. Since mRNA levels do not always correlate with functional protein levels, pairing single-cell RNA data with single-cell protein expression data gives you more accurate and complete conclusions about cellular function. In this sample, all cells are positive for HIF1-alpha RNA, but Milo shows that only about half of the cells contain HIF1-alpha protein. Milo users are uncovering novel insights at the single-cell level that are leading to publication in the top scientific journals. Here's how it works. You put your cell suspension on the SC-West chip and put the chip in Milo. Milo captures over 1,000 cells, lyses them, runs an SDS page separation on every single cell lysate, and immobilizes all your separated proteins in just 5 minutes. Next, you probe your protein targets on chip with standard primary and fluorescent secondary antibodies, and then image chip fluorescence. Data analysis is then performed for you automatically using ProteinSimple Scout software. The entire process from sample loading to data analysis takes just 4 to 6 hours with no overnight transfer step. And you can use off-the-shelf primary Western antibodies, so you can be sure you'll find an antibody against all your targets of interest. Here's what Dr. Maya Kilbear, a postdoctoral fellow at Duke University, had to say about her experience using Milo to uncover new insights she published in Science about how the gut and brain communicate.
In our lab, the way that we've been using Milo is to pair it with kind of single-cell sequencing data. So we have the RNA, we know the transcripts are there, but this is actually a way for us to measure and quantify the amount of protein that's in each individual cell. My favorite feature of Milo is definitely how easy it is to use. You basically just put your cells on their slides and pop it into Milo. You run the gel for a short period of time, right? It's just seconds. And having that automated as opposed to manually trying to turn on and off things, it made it a lot easier. The power of it, which I really liked, is the fact that you can assay a number of cells in these kind of micro wells and then get their protein output. Yeah, so the software is very easy to use. It's fairly intuitive, which is pretty much what you would want from your software, and it tells you exactly what you need to know. I would recommend Milo to my colleagues, and I have actually discussed it with some of them, and we are moving forward with working with it on our next study.
To request a quote and learn more about Milo from ProteinSimple, click the button below.